Welcome everyone to our presentation. Today I, Rishita Agarwal, alongside my teammates Clara and Sarah Chretien, will be exploring carbonaceous meteorites and their role in the origins of life. Our current understanding of the events that led to the origins of life on Earth remains limited, especially during the time frame between 4.5 and 4 billion years ago. So, in an attempt to shed light on how life might have emerged during this period, we extensively reviewed numerous online scientific studies and articles with a focus on carbonaceous meteorites and their composition. Approximately 4.1 to 3.9 billion years ago, the late heavy bombardment period occurred, characterized by a greater frequency of asteroid impacts throughout the solar system. An intriguing aspect of the late heavy bombardment period is the participation of carbonaceous chondrite meteorites of the CM2 type. Meteorites of this type have been found to contain a rich mixture of complex organic compounds, including proteins such as amino acids, as well as purine and pyrimidine nucleobases that contribute to the formation of RNA. This finding is significant because, according to researchers, the initiation of life requires the presence of RNA and proteins. Furthermore, the earliest signs of life discovered by scientists date back to around 3.7 billion years ago, which is relatively close in time to the late heavy bombardment period. This all hints towards that the late heavy bombardment period had something to do with the beginning of life. And so, this project specifically aims to investigate how carbonaceous chondrite meteorites could have contributed to the emergence of life during the late heavy bombardment period.
We determined our best approach would be to study the compounds found in carbonaceous chondrites, assuming ancient carbonaceous chondrites had a similar chemical makeup to the meteorites found in recent years, meaning organic compounds such as amino acids, alcohols, and sugars could have been delivered to early Earth. Specifically, amino acids and nucleobases have been found in CM2 meteorites such as the emergence meteorite. The emergence meteorite contains glycine, alanine, valine, proline, and glutamic acid, which are all protein-making amino acids. Scientists also found purines and pyrimidines on Murchison. Moreover, some CM2 meteorites contain the 5 RNA-DNA component nucleobases. They contain around 300 parts per billion of adenine, guanine, and around 50 parts per billion of uracil, but initially no trace of cytosine or thymine. However, after realizing the formic acid used to extract nucleobases was dissolving cytosine and thymine, scientists began to use cold water instead. This change revealed the presence of cytosine and thymine in CM2 meteorites such as the Murchison meteorite and the Tagish Lake meteorite.
Moving forward, research may be conducted into late heavy bombardment impact events on Mars, where meteorites, particularly carbonaceous chondrites, may have deposited organic compounds. Research should also be focused on the Moon, where previous Apollo mission samples have identified carbon compounds, particularly amino acids, in lunar soil. However, it is important to realize that these samples may have experienced contamination either through rock exhaust or solar wind, and not particularly through impact events. Nonetheless, identifying the presence of organic compounds in relation to meteorites on the Moon and Mars may suggest a relationship between the distribution of organic compounds on the aforementioned celestial bodies and Earth, which in turn would support speculation that it is standard for organic compounds to be distributed through carbonaceous chondrite impact events. Efforts should also be made into determining the process of complex organic compound formation in space versus on terrestrial planets. Through the Miller-Urey experiment, scientists Miller and Urey demonstrated how early Earth conditions could produce complex organic compounds such as amino acids, though the presence of organic compounds in carbonaceous chondrites is far more uncertain. Furthermore, because of the Miller-Urey experiment, it can be inferred that unless the rate of complex organic compound formation in carbonaceous chondrites is significantly higher than through the Miller-Urey process, then carbonaceous chondrites would not have had a meaningful impact on the distribution of complex organic compounds and subsequently life on Earth. Further research should also be done into the presence of RNA nucleobases in CM2 meteorites and whether RNA's other components, ribose and phosphate groups, also tend to be present in similar meteorites. The greater the number of instances of CM2 meteorites with all 3 components of RNA, the more plausible that meteorites played a role in the formation of RNA on Earth and influenced the evolution of life. Geologists finally should develop a stronger picture on the frequency and intensity of light heavy bombardment impacts by studying spiral layers. Spheral layers are sedimentary beds containing spherules, or deposits of meteorite impacts formed from ejected impact melt or rock vapor plume condensates. They serve as markers for ancient impact events and can be used to study the frequency and intensity of impacts during certain time periods. A high frequency of impacts during the late heavy bombardment period would support the theory that the origin of life was influenced by impact events.
We investigate the potential relationship between carbonaceous chondrites and the origins of life. With this objective, our team learned of organic compounds found in CM2 meteorites. Amino acids and nucleobases were found to be the most relevant in our research. These monomers for proteins in DNA, RNA, both of which are essential to cell structure, lead scientists to believe that either compound was the first organic molecule on Earth. RNA carries the instructions for cells, and proteins are the material used to build cells and fulfill these functions. The existence of these organic compounds within meteorites indicates that organic molecules could have been produced abiotically and delivered to Earth during the late heavy bombardment period. These compounds could have provided Earth's source of amino acids and nucleobases that then evolved into early life. Based on this information, we believe carbonaceous chondrites, specifically CM2 meteorites, brought the components needed for early life to Earth.
SEAS has allowed me to connect with so many accomplished professionals who dedicated their whole life to the sciences. Their talks were so interesting and have inspired me to really continue pursuing my interest in astronomy.
This experience has allowed me to meet with NASA scientists in fields that I'm interested in and hear about the different paths they took in their careers. This will help me plan out my future education and career.
My experience with SEAS has educated me on how to conduct research projects and presentations. Through this program, I feel more prepared for my future endeavors. We would like to thank Margaret Baggio, Selena Miller, the University of Texas at Austin Center for Space Research, Suzanne, Foxworth, and Maritza Montoya, all the NASA Aerie speakers, and all the SEAS speakers. Thank you so much.